Warahmatullahi wabarakatuh. Today I will explain about the semen analysis and sperm function capacity. We're going to learn about the morphology first because you can see that the sperm cell it has a unique shape and it is like different compared to the other cells. And let's start with the head first. So the head you can see in here it contains acrosome and also nucleus. Well, the acrosome is the one that is becoming the drill or like enzyme drill that will penetrate the ovum. So when like they are when the sperm want to go inside of the ovum, uh the acrosome is the one that is the front that will go in. And we also have nucleus. Like usual, nucleus is the one that have the genetic material or like genetic information. And then for your information, so the acrosome is actually the modifications of the lysosome that was formed by the vesicle aggregations from the Golgi and also the endoplasmic reticulum complex. And this area acrosome, which is like in the tip of the head, they have like a clear color, and also it is like covers 14 to 70% of the head, like 40 until 70% of the head, and they also have no organelles. Okay, so because of that, we have some kind of abnormalities if the shape is very small, or like the shape it is like irregular, and also like containing chromatin, vacuoles, or septa. Okay, like we know first, like it doesn't have organelles, but if they do, then it means it is considered as abnormal. And also the chromatin condensation, which is usually behind the acrosome. I don't know how to draw, but I think in here, around here. Well, they usually have the darker and also like homogeneous, and the abnormalities that happen because of the chromatin is that if they have some kind of like clumps of chromatin or like scatter chromosome threads. Okay, I think I need to add also for the head, we also have some kind like clinical like something that happened with the head abnormalities. The first one, if they are two head or like microcephalic, is usually associated with the polyploidy. So this one, which is mean like extra chromosome. Yeah, this one pas I mean like extra chromosome, and they have like poor fertility. And also for the small head or microcephalic, because lack of DNA or incomplete chromosomal packaging. Chromatin packaging. And also we have piriform or like pear shape, is often seen in varicocele or after the heat stress. So yeah. And then we have also the vacuoles in the head, which is like more than two small vacuoles or like large vacuoles that covers more than 20% of the head area. It could indicate the DNA damage, and this is also assessed with strictly under high power oil emission, like 1000 times. And then let's go to the neck part. You can see this neck is actually very small. So they are the one who connects the head and also the tail, which includes flagellum base and also like nuclear indentation. And this one is the one that connecting the plate with proximal and distal centrioles. And because it is like very small and very short, the abnormalities that happen in the neck is usually like grouped with the mid piece abnormalities in here. Yeah, because mid piece is actually already part of the tail, so that's why, because the neck is very small, if something happen with that, it's actually include with the mid piece as well. Okay, why the neck is very critical is very important, because the proximal centriole in here, yeah, is the one who initiate the first mitotic division of the zygote after the fertilizations. So if we have like a defect neck, it can cause a fertilized egg to fail to develop. Okay, and then let's go to the tail, which is like the longest one. It has like 9 until 10 times of the head length. It could be straight or like wavy, and it is also made into 3 parts. The first one is the mid piece, and then the principal piece, and also the end piece. Okay, it says that the mid piece is the energy, because where the energy is produced, because what? Because they have mitochondria in here. Okay, so this energy is the one that gives the energy, like the energy to move, for the motility. And for the principal part, like from here and to here, this one is the principal one. This one is like the longest one, which is with the fibrous sheet. And also for the end sheet, and the end piece is the terminal one, so like the last one. And yeah, like the structure is like a regular cilium or cilia. Okay, and then for the tail abnormalities, it could be from the length, if it's like less than 40 micrometers, or like the shape is not straight, or like too wavy, or like bent, or like broken into 2 or more parts. And also if they have like multiple tails, like more than one, is usually, if they have this one, usually they also goes with the head abnormalities. And for the other abnormalities is because of the incomplete growth. So yeah. And then I think I need to add also, if the tail defects, so like if they are like stiff or like coil tail, I think they have the oh no. Okay, so like for that one, if like stiff, how can I say like this, stiff or like coil, is usually because of the cold shock, or like pH is abnormal, and then the epididymis the pathology. And also if the tail is absent, of like there is no tail at all, or like it is like too short, it's usually because of like genetic defect that cause complete immotility, so like they cannot even move at all. And for the broken mid piece, if the mid piece in here is broken, they also cannot move, because the mitochondria is lost, so like they cannot have energy to move, right? Okay, and then next one we have the cytoplasmic granules or droplets. So this one indicates the impaired maturation. Well, it could be on the head, mid piece, or along the tail. Even if the morphology is normal, well, the presence of a droplet is usually considered as abnormal. Because what? Because the small droplet at the mid piece is normal in the sperm from the epididymis. So if just like from epididymis they exist, it's normal. But when they being ejaculated, they should be absent. So like they doesn't have anymore, like for this one. And the droplets is more than 1/3 the size of the head, are like significant abnormalities as well. Okay, next we have spermatogenesis, or the sperm making. So this one you can see. So the spermatogenesis is where the relative undifferentiated primordial germ cells, or spermatogonia, which it deployed 46 chromosomes that was being proliferate and transformed into highly specialized and motile sperm, which each with haploid 23 chromosomes. And well, this is the process, but I think you don't need to know like too much about this, because it's not the things that we are going to learn. I mean, you have learned this in the biology, so fine. Let's go to oh yeah. Oh yeah, I think I need to say this. So like if like the man is having like any toxin, fever, or like medication that will affect. So like this, okay, this one spermatogenesis has happened like in total like around 74 days. So like if someone is like having like fever, medications of anything, it actually could affect the sperm quality in like around 2.5 until like 3 months later. So like if I get sick, I mean not me, if like a man gets sick today, so like in the next 3 months he could have some kind of abnormal morphology or like motility of the sperm. Okay, let's just go to the next one, which is the specimen collection. Well, I don't think I need to explain this too much, but I just going to add some of the things. Well, why they need the probandus to be abstinence or like didn't do any ejaculations for at least 48 hours and until 7 days and not more than that. Well, because if like the abstinence is shorter than 48 hours, it could have like lower volume but higher mortality. Okay, so like the shorter abstinence, okay, means that the volume is volume is lower but higher motility. But if it's like longer abstinence, like longer abstinence, more than 7 days, blah, the higher the volume means the more dead or abnormal sperm. Okay, and then it says also like they need to avoid using some kind of like lubricants, saliva, or like soap, because all of those things could make the sperm dead. And also it says that we need to protect them from like extreme temperatures. Like the ideal temperature is like 20 until 40 degrees Celsius, but if it's like below than that, it could have some kind like below that. Below 20 Celsius could have like cold shock, but if it's like higher than 40, it could kill the sperm as well. Oh yeah, for the cold shock, it could make the sperm cannot move at all. Okay, I mean they're still alive but they just cannot move. And because this specimen is very, I can say like dangerous. I mean like it could transmit many things like disease, like hepatitis virus, HIV, and herpes and others. So that's why you need to wear the gloves and also the lab coat. It's going to be better if you work in a biosafety cabinet, but in the physiology lab doesn't have that one. Okay, let's go next with the microscopic examination. So microscopic, it starts with the liquefaction. Okay, so liquefaction is usually happen like 30 minutes until 2 hours after the ejaculation. That's why the probandus need to do the ejaculations right, like 1 hour before the practicum is starting to prevent the dehydration, and also the temperature changes. So for the macroscopic, we have several examination. So it's macroscopic. The first one is this liquefaction. So you know that the cement is actually like semi-solid clot because it is like due to the seminal vesicle coagulum, but within like some minutes at the room temperature it could be like thinner, or like, yeah, they get thinner, and it is usually complete in like around 14 minutes or until like 1 hour. If it's not liquefied, okay, if not liquefied more than 1 hour, it could be that you need to note this because it could be it has some kind of like abnormalities, such as dubelcose phosphate buffer saline, but I think you don't need to know that. Oh yeah, the failure of to liquefy usually suggests to prostate dysfunction. So like fail to liquefy means that prostate dysfunction, but if it's like they are too fast to liquefy, it could be that there's some like infection happen, or infection, because like infection will release the leukocytes protease. And then it says there. Okay, it's like less than 5 minutes. The next one is actually the volume for normal one. Wait, it doesn't have it. Normal, if I'm not mistaken, it's like 1.5 until 5.5 milliliters. So if it's like low, it is because of the retrograde ejaculation, or the ejaculatory duct obstructions, or hypogonadism. But I don't want to write it again because it takes a long time. And then if it's like higher than 5.5, it could be because of the dilute sperm concentration. Okay, let's go to the color. Okay, usually the color is like the gray opalescence. So if color gray opalescent, if it's okay, if the color is like less opaque, it could be that the sperm is low. If the like brownish red, it could be that they have like RBC. So it has also the name which is called as the hemospermia. I mean, hemo means blood, and this one is sperm. And then we also have, oh yeah, this brownish red colors is also like indicates anything like infections or like tumor. And if it's like yellow, could be because of vitamins or like medication, or high leukocytes because of like piospermia. I don't need to tell this. Okay. And then viscosity. The first one is viscosity. Viscosity is to see like how can I say? Yeah, the thickness. So you can like using the pipette and then you drop like one drop, and with that you need like, it has some kind of like string, and you need to measure that. If it's like more than 2 centimeters, it is considered as the abnormal, or like high viscosity is because like traps sperm, which is could reduce the mortality if the sperm are healthy. And this one is also can be due to the prolonged abstinence or infections. And wait, and then odor is the smell. Usually it's like acacia flower. Well, it's because of the spermine and spermidine. Well, the smell, it could be like bad smell. It could be from like pus or like infection, something like that. And then the next one, the last one is the pH. We just like need to put like the pH paper and weight. The normal is 7.2 until 8. If it's like too acid, it's because of the abstinence. The absence of seminal vesicle fluid. Or like obstruction. But if it's like too alkaline, it's because of like acute infection or like prostatitis. Okay, that's done for the microscopic examination. Let's go to the microscopic one. If microscopic, it means that they need microscope to see it, and they will provide you this one. For the first one is agglutination. So agglutination is to see whether the sperm is like sticking to each other, like head to head or like tail to tail, or like mix, which could be indicates of the anti-sperm antibodies. So the first one is agglutination. Okay, this one they have some kind of degree as well in here. I don't need to write it again. So if it's like isolated, it means like there's a lot of sperm that is just free, they don't mix to each other. And if it's like moderate, there's some like they are being agglutinated, but there's some also like the free sperm, so like could be like 50-50. For the large one, it means that the agglutination sperm is more than the free sperm, but for the gross one, all the sperm is being agglutinated. Okay, and then the second one we will going to do the motility. Yeah, motility. Sorry, motility is to see the movement of the tail. So, okay, wait. Okay, we're just going to go into the categories. Okay. The categories, they also have like 4 types. The first one is the rapid progressive, or like PR, and also progressive. It usually rapid progressive. It means that they are fast and also like straightforward movements. So like they just go straight. It doesn't go to like left or like right. And then we also have the slow progressive. Well, for this one, they are still move, but it's just not straight. But their movement is also like slow or like difficult. And the third one is non-progressive. If they are moving but they're just stay in place, like I mean like their tails is move, like yeah, they're doing some kind of move, but they doesn't move to that place. And then the fourth one is if they are do not move at all, just immotile. It means that usually it means they are dead. But okay, to see that one, we need to do the other examination, which is called as, wait, I forgot. Okay, let's go to the next one, which is HOS. HOS means hypoosmotic swelling test. So, if the membrane of the cell is good, it means that, okay, I think I will explain this. If the membrane is good, then solution will enter the cell and the membrane will swell. And the tail will coil. It could be like the coil will be like only the tip, or like the third, it could be like half of that, or could be like full of them are coil. So it says that if it's more than 60% are positive or like coil, it means it is normal. But if it's doesn't do that, it could be that the sperm are not alive at all. So like for this one, usually to see if it's okay. Oh sorry, I think oh no. Okay, just go with this one. Okay, the fourth one is the viability. Where's viability? Oh yeah, this one. So to see if the sperm integrity, okay. So they have actually like eosin and also nigrosin. I mean like the solution that they need to drop. So if the result is being had, if it's being had, it means that the sperm is dead. Sperm is dead because the membrane damage, so like the dye is entered. But if it's like just white, it means they're still alive, because like the membrane is intact and exclude the dye. So if the fiability is like high, like more than 60%, but the mortality is low, like less than 40%, it could suspect into the structures of detailed defects. So that's why they are immotile cilia syndrome. But if both fiability and mortality are low, we could suspect them into necrozoospermia, or like, I forgot what is the meaning of that. Okay, let's go to the count test. Number what? Number 5. Count test. Count test. Well, they have the formula in here. So like the one that you find, you count is times with 1,000,000, and you will get the result. And yeah, I mean, like, you can see it, the result. And then, okay, this one is the normal value that you can remember, because if I'm not mistaken, from last year, usually in the pretest they will ask about the normal value, and then also like what are the tests that was being implemented in the microscopic, like you can please like name them all, and then the microscopic, what are they, and then what else, like this one, the acrosome, like what is the functions of the acrosome, and also like the acrosome is the modifications of what, and then what else, if the head is being abnormal, what are the lists that we can, I mean, like it can be like, for example, like the shape or like the size and others. And if the tail is also having like abnormalities, what are they? And also, I think those are just the one that will be asked in your pretest, and it's not that hard. And thank you.